Welcome everybody to today's webinar. Thank you so much for joining. I will talk about setting up highly efficient cell line development workflows. My name is Ellie and I'm an application scientist at Cytina. This presentation will last about 20 minutes and there will be a Q&A session afterwards. I want to briefly introduce who we are and what kind of products we are developing. Since last year, we're now a part of a larger corporation, Cell Inc., which was founded in 2016 and has grown rapidly. By now, we are present in over 60 countries and have offices all over around the globe, develop new products, and support our customers. This drive for innovation enables us to significantly grow our product offering, which offers a number of different areas within life sciences. Today, we offer solutions across different areas of work, including bioprinting, liquid handling, and analysis. At Cytina, we developed instruments for automated isolation of single living cells since 2014. All our instruments are capable of label-free cell isolation and come with image-based assurance of clonality. It started off with the single-cell printer in 2015, and more recently, we launched our Xite series, which consists of the 3 benchtop instruments for isolation of mammalian and microbial cells. Introduced this year is the CBIRD, which is a device that enables early suspension mixing, available in both 96 and 24 well plates. If you look at the largest pharma companies, most of them already are our customers and have purchased at least one of our instruments. For us, this is really great to see both the acceptance and adoption of our technology in these companies. So this is a brief agenda of today's webinar. After introducing the most prominent applications that are addressed by our customers, I will explain how our single-cell dispensing technology works. Then I will discuss important aspects of single-cell cloning in cell line development, and also share one of our most recent developments in this webinar, the CBIRD, which is a novel well plate-based microbioreactor for early suspension culture. I would like to provide a short overview on the applications we focus on. Our instruments are already well established in CLD. There are a lot of details and also variations, but from our perspective, the workflow starts from transfection, followed by single-cell isolation. Assuring clonality is important to meet the requirements of the FDA and other regulatory bodies. Growing colonies then need to be selected based on productivity, product quality. Further, the stability of the cell lines need to be guaranteed before one can select the final production clone. There's also 2 more applications outside CLD that are well suited for our instruments. Here, this includes the cultivations from mixed samples, which follows a similar process, including single-cell isolation, and also single-cell omics, which includes single-cell genomics and transcriptomics. And now we offer liquid handling to better miniaturize and automate these workflows. But today I will focus on CLD. What are the major challenges in cell line development? Having worked now for several years with some of the leading CLD groups, it boils down to 4 major challenges. The first challenge is the assurance of clonality, which is also a regulatory requirement. The second one is clone recovery. Downstream of single-cell cloning, the major challenges are cell line stability and productivity as well as quality. These are key parameters that have to be guaranteed for the final production clones. The current gold standard for assuring clonality is automated well imaging. This technology, however, is not error-free. It can be frequently seen that 1 to 5% of cells are deposited into the well plates and are not detected by the imager due to artifacts or edge effects. This can also lead to so-called ghost wells, where there is no cell detected, but a growing colony emerges. Today, I will talk about 2 technologies that address some of these challenges. Most importantly, single-cell dispensing, which allows for deterministic cell isolation with image-based assurance of clonality and results in high cell viability. The second technology is plate-based microbioreactor for early suspension mixing, which better reflects the final culture conditions in a microbioreactor after single-cell cloning. Our instruments provide assurance of clonality with no cross-contamination and high cell viability. So this is solved by our single-cell printer, and here's an image of also our cartridges. So With our C-site, this is an instrument that is for label-free isolation of mammalian cells. It is a benchtop instrument that comes with the software that is super easy to use and user intuitive. If we zoom into the printhead here, you can see the nozzle of our disposable dispensing cartridge. We use an optical detection system similar to a bright field microscope to detect cells in the nozzle of our dispensing cartridges. Only those droplets that contain a single cell is deposited into the target substrate. To dispense the cells, the is similar to an inkjet printer. So first you have the droplet ejection, where the piston moves inward, displacing liquid, and the droplet breaks up, and the droplet is released. Then, when the piston retracts, new liquid is drawn into the chip, and here this is the droplet release. Droplet on-demand system has very low shear forces compared to other dispensing technologies. With the next few slides, I will explain the single-cell dispensing principle in some more detail. So the automatic detection algorithm analyzes the nozzle area. In this case, there's no cell present, so the shutter is set on, and the ejected droplet is sucked away. All the cells in the chip move closer to the nozzle. So now we have a new situation, and there's a single cell in the nozzle. Here, the shutter is shut off, and the single cell droplet is dispensed into the well. The algorithm is also capable of measuring a variety of morphological parameters, such as cell diameter and roundness. Therefore, it can even detect two cells sticking to each other and send them to waste. The shutter will then remove the droplets with multiple cells or particles that don't fit the morphological criteria set by the operator. So in the X-Site series, we have the C-Site that you just saw. We also have an instrument with a fluorescent detection channel, the F-Site, and an instrument for isolating microbial cells, the B-Site. All instruments use sterilized disposable cartridges to prevent cross-contamination, which come in packs of 10, individually wrapped. Since this is the only part that gets into contact with the sample, there are no routine cleaning procedures required. For each cell that is dispensed, several images are stored on the hard drive to document the cell isolation. This is one example how the nozzle images that we store for assurance of clonality look like on the F-Site. This demonstrates the working principle very nicely. Here you see a single GFP transfected CHO cell a little bit above the nozzle. To prevent premature bleaching, we only illuminate the nozzle area with blue excitation, which is around about this area indicated. So here, there's currently no cell in the nozzle, so the system dispenses an empty droplet, which is automatically sucked into waste. Now the cell moves into the nozzle, and you can see that it fluoresces. The image processing result is also saved with an overlay. And you also get an image of the nozzle after dispensation to show that the cell has actually left the nozzle. Many of our customers in the pharma industry include these images to prove clonality of their production cell lines to the FDA or other regulatory bodies. One exciting application of the F-Site that I would like to highlight is cloning of CRISPR-Cas9 edited stem cells. Our partner at the University of Freiburg used our instrument to isolate rather delicate cells after transfection and genome editing. The F-Site helped a lot to simplify this workflow, especially since small sample volumes were available after transfection. As you can see, with up to 40% clone recovery, they reached very decent outgrowth given that these are rather delicate cells. When it comes to what can be achieved with respect to assuring clonality with our technology, I like to refer to this publication by Mandy and David from Genentech. They actually established a fully automated workflow with a single cell printer. In their validation round with over 3,000 clones, they achieved a single cell isolation efficiency of 96%. This paper provides a detailed description of how thoroughly they assessed the probability of clonality. They demonstrate that using the state-of-the-art plate imager in combination with the SCP results in a probability of clonality that exceeds 99.99%, and therefore clearly meets the common requirements for the regulatory bodies. They also state that the SCP results in dramatically reduced number of plates needed for cloning compared to other automation workflows. If you're interested in learning more about the assurance of clonality, the pros and cons of plate imaging, and appropriate metrics, I can recommend these two publications. A bunch of our customers are among the authors, such as a group from Amgen. Please shoot us an email if you're interested in the original publications. So here is a brief summary of the key features of our Xsight instruments for CLD cloning. Essentially, we have image-based assurance of clonality, easy to use benchtop instrument, of course, no cross-contamination because we have disposable cartridges. We achieve a single cell efficiency of greater than 95% with high cell viability. Our dispensing time is very fast with 2 to 5 minutes for a 96 well plate and 10 minutes for a 384 well plate. And we're also automation ready. So in the second part of the talk, I will introduce to you the CBIRD system, which is one of our latest developments. It is a system that brings suspension mixing culture to the very beginning of the CLD process and can be rapidly integrated to common CLD workflows. So in traditional static culture, cells are limited to a 2D growing space. Oxygen comes from the gas-liquid interface, which is far away from where the cells sit. All the waste metabolites that the cells produce are concentrated around the cells, and this makes for an unideal environment for the cells. If we enable the mixing culture, the suspension cell lines now have a 3D growing space, and all the cells have an equal distribution of the oxygen and nutrients. Even for adherent cells, this can also refresh the media around the cells as well. So here is a cell line development process published by well-known pharmaceutical companies. Cell line development process typically starts from single cell culture in 96 well plates and continuously scales up to 24 well plate, 24 deep well plates, and shake flask. As you've already noticed, that mixing of cell culture only begins in the shake flask or 24 deep well plates in all the later stages. However, in the first two stages, the 96 well and 24 well plates, current technology in the market does not enable mixing culture. Therefore, pharma companies can only do static culture, which is not ideal for the cells. And this is how we introduced our CBIRD. It's a way to fulfill this gap to enable the suspension mixing in 96 well and 24 well plate stages. Our CBIRD system consists of a docking station and 3 control units, which can operate 3 96 or 24 well plates. The CBIRD working principle is very simple. There is a CBIRD lid, which can fit with the standard 96 well plate. The lid contains 96 tubes, which are inserted into all the wells of the plates. And the CBIRD control unit is docked right on top of the CBIRD lid to pump the media up and down in all the wells, somewhat like a multi-channel pipette system. In this video below, we have placed a camera below the well of a CBIRD, so you can see that the CBIRD mixes the suspension of the CHO cells up and down. I would like to point out that CBIRD enables customizable mixing options. While mixing is generally good for cell growth, too much mixing may be detrimental for low cell density. Therefore, for low cell density culture, say from a single cell to about 10 to the 3 cells per ml, we usually recommend periodic mixing. And then from 10 to the 4th to 1 million cells per ml, we recommend continuous mixing of cell culture for most optimal growth. The mixing conditions can be tailored to your needs and also the cell types. In this experiment, we look at cell growth and protein yield of CHO-K1 cell line that produces HER2 antibody that's used for the treatment of breast cancer. We compared CBIRD to static culture in 96 well plates and took the endpoint measurements. Case 1, we seeded 500,000 cells per ml and cultured for 5 days. In case 2, we seeded 1.5 million cells and cultured for 3 days. As you can see from the charts on the right, the CBIRD consistently produced much higher cell density and antibody protein yield compared to static culture in both case 1 and 2. The CBIRD also produces higher QP value compared to static culture. So here we demonstrate that the CBIRD provides more optimal cell growth for higher cell density and protein yield. So the second data set was generated by our customers. They seeded cells at 3 different densities and compared seabird and static culture for cell viability and density after 3 days of culture. At low densities, the seabird does not greatly differ from the static culture in terms of cell viability. However, at higher cell seeding densities, seabird enables much higher cell growth, up to 5 million cells per ml, with 98% cell viability. Static culture only has 1.3 million cells per ml with 84% viability. Here, the seabird can stretch the growth potential of cell culture when static culture reaches its limit, and the seabird can push it to higher cell density. So we also did a correlation assay to see how well seabird compared to shake flask conditions. We compared seabird and static culture to 125 ml shake flask with 30 ml of cell suspension. We measured cell density every day for 5 days, and you can see that the cell density of seabird and shake flask group are similar, but the static group had a lower density. This shows that cells growing in seabird conditions mimics the growth rate of cells grown in a shake flask. The same goes for doubling time and titer of protein yield after 5 days. Seabird culture conditions are much more representative of latter stage shake flask conditions. This is extremely important for early selection of clones, which saves time and cost for the CLD process. We also measured the oxygen value at the bottom of the well. The seabird increases oxygen levels at the bottom compared to static conditions. This is important to prevent hypoxia in cells. We can also show that the seabird has no cross-contamination issues. One of our customers tested this by intentionally seeding cells only in rows A, C, E, and G, and continuously cultured for 7 days. After 7 days, they only saw the cells in those original rows and did not see cells in rows B, D, F, and H. At the same time, they also measured the titer production, and again, the seabird achieved a higher titer than static culture. So with that, how can we come together? So we're very happy to set up an individual telecon to discuss your applications and workflows. After such telecon, we typically proceed with the on-site demo to introduce you to the instrument and work with your cells and study efficiency and growth. Of course, with this situation with COVID-19, there's severe restrictions in travel. So therefore, we've set up a live demo lab to offer online demonstrations on request. So if you're interested, now's the chance to get an application-specific live demo so you can watch from anywhere. Please get in contact with us, and our team is ready to prep the demo for you. And with that, I would like to thank you so much for joining me today. I hope you enjoyed the presentation, and now I'm ready to take questions. Thank you.